Hey, welcome back everybody. Again, my name is Nick, and in this video, we're going to go through the high-yield information surrounding plaque and biofilm. Okay, here we have some visible dental biofilm on the surfaces of these teeth. Biofilm can also be called plaque, and it's a group of bacteria that sticks on tooth surfaces. It includes not only the bacteria, but their byproducts and a sticky matrix called an EPS. EPS stands for extracellular polymeric substance. So the EPS is what gives the biofilm structure and protects the bacteria. Now, a key bacteria you should definitely know as a dentist and for the exam is Streptococcus mutans, and it's strongly linked to dental caries. This bacteria makes an enzyme called glucosyl transferase. This is an enzyme that turns sucrose into dextrin, and dextrin is this sticky polymer that helps the bacteria stick to the tooth and group together. Of course, Strep mutans is not the only bacterial species in biofilm. Each biofilm contains hundreds of unique species, and we'll learn about some throughout this video. Bacteria found in biofilm are much harder to kill than bacteria that live on their own. So biofilm gives them a stable environment, makes them more resistant to threats like antibiotics. So I want you to know 4 specific ways that biofilms help bacteria. The first is by supporting a broad habitat. So biofilms support many types of bacteria, both aerobic and anaerobic, depending on where it forms in the mouth, specifically in the sulcus. We'll learn about that in the next few slides. The second thing I want you to know is metabolic diversity. So biofilms are metabolically active and very efficient. Bacteria in biofilm share nutrients with each other. One species can have a byproduct that they don't need, and guess what? That's another substrate or a different species. So it creates this symbiotic food chain and improves efficiency since there's no wasted nutrients. And here's one twist students often miss. Even though biofilms are metabolically diverse, many of the bacteria deeper inside the biofilm actually shift into a low metabolism or a slow growing state, and this makes them much harder to kill with antibiotics, which brings us to our next point. The third is that biofilm are the most resistant. So there's enhanced resistance here. Bacteria can share resistance genes. And as a refresher, this is called horizontal gene transfer. So this includes genes for enzymes like beta-lactamases, which can actually break down antibiotics. So the biofilm can also block antibiotics from reaching the deeper parts just because there's not enough penetration. Biofilms also protect themselves by eliminating competing microorganisms. They release acids, toxins, bacteriocins that suppress or kill species that threaten the biofilm's stability. So only the bacteria that support the community survive. The last thing I want you to know is that there's increased pathogenicity with biofilms. Biofilms lead to chronic infections like periodontitis and cavities. That's because they boost the production of harmful toxins, acids, and proteases that can damage the surrounding tissues. Biofilms are the main cause of periodontal disease and how our immune system reacts to that. So biofilms are found in both healthy and diseased mouths. The difference is the type of bacteria that they contain. So healthy gums have healthy bacteria, and as such, they show clinical health. In gingivitis, the gums are inflamed, but there's no permanent damage to bone or the periodontal ligament. There's no increases in probing depths. Periodontitis is more serious. It involves permanent destruction of the periodontal ligament, alveolar bone, and surrounding tissue. So let's take a quick look at the high-yield points surrounding healthy gingival tissues. So in healthy gums, the sulcus mainly contains aerobic gram-positive bacteria. That's the take-home point that I want you to remember. The most common types are Streptococcus and Actinomyces species. These bacteria are non-motile, meaning that they don't move on their own. They just stay in place, and they don't have structures like flagella to help them move. They're part of the normal microbiome, help keep the gums healthy. So now let's take a look at gingivitis and see how that's different. So as plaque builds up, the oxygen levels drop in the sulcus. So this creates an anaerobic environment. More gingival crevicular fluid, or GCF, also flows into the sulcus during inflammation, and these changes cause the bacteria to shift from an aerobic gram-positive state to a anaerobic gram-negative composition of bacterial species. So in early gingivitis, the first immune cells to respond are called PMNs, or polymorphonuclear leukocytes. This group includes neutrophils, eosinophils, and basophils. Neutrophils are the main ones in this stage. A high-yield condition surrounding gingivitis to know is necrotizing ulcerative gingivitis, called NUG. Sometimes it can be acute. It's called acute necrotizing ulcerative gingivitis. So you may see NUG or ANUG. And this is an infection with the following signs. You have hunched out papillae, gingival bleeding, pain, may have a fever, and there's a nasty bad odor to it. But what's important here is there's key bacteria in necrotizing ulcerative gingivitis, and that is Fusobacterium. Prevotella intermedia, Treponema denticola, Porphyromonas gingivalis, and Tannerella forsythia. Lastly, let's take a look at periodontitis. So periodontitis, as we know, is a disease caused by several factors working together. These include bacteria in biofilm, and the body's immune response to that, and the environment. So the main idea in periodontitis theory is that we control the disease by managing the body's immune and inflammatory responses, not just killing the bacteria themselves. This is because tissue damage and bone loss happens mainly due to an overreactive or faulty immune response. In terms of immunology, what I want you to know is the most important immune cells are plasma cells. So plasma cells come from B lymphocytes, and they make antibodies to fight the bacteria, but their constant activity causes ongoing inflammation, which can damage other tissue and bone. You may run into a process called epithelial shedding. This is where the top layer of cells in the gingiva naturally sloughs off and is replaced by new cells. So this helps protect the gums by removing bacteria that are attached to the surface and stopping them from going deeper and infecting the tissue. And then another key point you need to know is that just like in gingivitis, periodontal pockets have more gram-negative anaerobic bacteria. Okay, here's a diagram that shows microbial complexes. So a complex is a group of bacteria that live and work together in their specific dental plaque. So they're grouped by how they behave and their role in health or disease. So I'm going to briefly go over all of these and talk about some high yield points. So the blue and yellow complexes are healthy. These are what are considered early colonizers, and they're linked to healthy gums. The next is the pink orange and the orange red complex. This is the transitionary complexes. So these appear as biofilm matures and becomes more harmful. The important ones that you should know are Prevotella intermedia. That's an easy one to remember. Intermedia, it's intermediate. Fusobacterium nucleatum, this is a key bridge bacteria, and Campylobacter species. And then lastly, and most importantly, we get to the red complex. This is what causes the disease. So these bacteria appear last and are linked to periodontitis. They include Porphyromonas gingivalis, Tannerella forsythia, denticola. Those are the three ones that you definitely need to know. There's a specific type of periodontitis called Molar-Incisor pattern periodontitis, and you're going to see this in a few other videos. It's sometimes called localized aggressive periodontitis historically. So Molar-Incisor pattern periodontitis, MIPP, affects the first molars and incisors. Easy one to remember. And this disease usually shows up only in teens and young adults. And the key fact here that you need to know is it's strongly linked to a specific type of bacteria, and that's called, get ready for it, Agrigidibacter actinomyces temcomitans. Yeah, that's a fun one to say. It's also abbreviated as just AA, and AA plays a big role in causing the damage here found in Molar-Incisor pattern periodontitis. Shifting gears here, let's look at Peri-Implantitis, and this is when an implant shows progressive clinical attachment loss. So this is caused by plaque that accumulates around the implant, and in response to that, we have inflammation, and as a response to that, we see bone loss around the threads of the implant. So it's similar to periodontitis, but happens around implants instead of natural teeth. Both Peri-Implantitis and periodontitis share many of the same harmful bacteria. That's the main point here. There's also bacteria that's unique to Peri-Implantitis, so it's not identical, but if you encounter questions about this, just know that the composition is very similar to periodontitis. The last thing that I want you to know is stannous fluoride, or SNF2. This is an important ingredient to know for its anti-plaque forming properties. It's in many different types of toothpaste. Stannous fluoride helps fight plaque, which in turn helps fight gingivitis. It also helps prevent caries because it has fluoride. So other fluoride types in oral care products are sodium fluoride and sodium monofluorophosphate. Those are good for caries prevention, but they don't fight gingivitis as well. So the key point is stannous fluoride helps with both caries and gingivitis prevention. Let's do some example questions here to help solidify our understanding. So in this question, which immune cells are commonly associated with early gingivitis and chronic periodontitis, respectively? Please pause the video and resume when you think you got it. The answer is B. PMNs, specifically neutrophils, are the dominant immune cells in early gingivitis. They're part of the innate immune system and act as the first line of defense against the initial buildup of plaque bacteria. In contrast, plasma cells dominate in periodontitis. Our next question is asking, which type of bacteria is commonly present in the periodontal pocket? Please pause the video and resume when you're ready. The correct answer is C, anaerobic gram-negative bacteria. Examples are P. gingivalis, Tannerella forsythia, Treponema denticola. These are all part of the red complex and they're anaerobic gram-negative. Here's a clinical scenario of a female patient who is 20 years old. Her chief complaint is that her front and back teeth are really loose. She has some family history of early tooth loss, and our current findings include localized probing depths of 6 to 8 millimeters in the first molars and mandibular central incisors. There is also radiographic vertical bone loss in the respective regions. So what bacteria is most likely responsible for this disease? Please pause the video again, and when you're ready, hit resume. The correct answer is D. The patient's clinical signs, namely deep pockets and vertical bone loss in first molars and incisors at age 20, are all key characteristics of molar-incisor pattern periodontitis. Option D, Aggregatibacter actinomyces temicomitans, AA, is the key pathogen in molar-incisor pattern, and that's what you need to know. Moving on to our last question, each of the following is a way in which biofilms create protective environments except one. Which one is the exception? Please pause the video, read through the answer choices, and when you're ready, hit resume. So this answer is A, high pathogenicity. That is not a protective mechanism of biofilms. It refers to the ability of microorganisms to cause disease, not necessarily its ability to survive in a protective environment. Okay, we made it to the end of the video, and here are some things that you should know for your exam. The first is that biofilms are highly resistant structures. Hopefully you appreciate that now. The second is that anaerobic gram-negative bacteria are what's found in periodontal pockets. Next, red complex bacteria are associated with periodontal disease. You should know the 3 red complex bacteria. Next, you should know about the bacteria AA and its association with molar-incisor pattern periodontitis. And lastly, you should know about stannous fluoride because it's anti-plaque and anti-gingivitis.